Discover how the Maryland Stem Cell Research Fund is accelerating cures.
Hello, my name is Jeff Bulte. I'm a professor of radiology at the Johns Hopkins University School of Medicine and serve as the director of the cellular imaging section in the Institute for Cell Engineering. As such, I specialize in molecular and cellular imaging and develop new contrast agents and imaging probes that can be used to locate and interrogate the fate of stem cells in real time in a living body without the need to perform biopsies or extract tissues. Why is this important? At present, once stem cells are injected in patients, we don't know where they go, how long they stay and where, and for how long stem cells survive. My lab aims to develop advanced imaging techniques that can help the treating physician to make informed decisions on the efficacy of stem cell therapy as related to the whereabouts of stem cells. Our work is not focused on a single disease, rather we aim to develop methods that are generally applicable so most patients can benefit from it. My first awarded Maryland Stem Cell Research Fund grant was on imaging of remyelinating stem cells in animal models of multiple sclerosis, and our lab was part of the first bone marrow stromal cell tracking studies in patients with MS and amyotrophic lateral sclerosis, or ALS, also known as Lou Gehrig's disease. A second grant centered around using mesenchymal stem cells for immunosuppression of transplanted human cadaveric islets in animal models of type 1 diabetes. This has led to the development of immunoprotective, alginate-based microcapsules that were able to prevent immune rejection by the host, which is a big problem with this form of islet cell therapy. The third grant involved a phase 1 clinical trial, and the goal here is to use fat-derived stem cells obtained from liposuction to treat breast cancer patients who have damage from radiation therapy and to image the persistence and survival of implanted cells with fluorine MRI. I'm happy to say that we have now received FDA approval to treat patients with our fluorine-labeled stem cells. With the next grant, we were able to image the biodegradation of hydrogels that are used to embed stem cells in order to improve their survival and escape from the host immune rejection by the central nervous system. Finally, the fifth and latest MSCRF funding has enabled us to develop a completely new imaging modality, magnetic particle imaging, as applied to remyelinating stem cells in an animal model of stroke. This should have an unprecedented detection, sensitivity, and specificity. So, in summary, our imaging techniques will assure that the doctors can see if they indeed injected the cells at the right place and stay there, so they know much sooner if the therapy is likely to succeed instead of finding that out when it may be too late. I want to emphasize that the overall funding by the MSCRF has allowed us to secure follow-up funds from other organizations, which otherwise would have been much more difficult. In addition, through the fellowship awards, I was able to hire and nurture many postdocs, several of which have now become successful faculty at Hopkins and elsewhere.
Accelerating today's stem cell research into tomorrow's cures.